Hello, my name is Jimmy Zhang, and I would like to thank you for listening to my presentations on the fundamentals of tissue engineering today. I will be covering the rationale behind the field of tissue engineering, as well as major components considered for the tissue engineering studies. Before I delve into the importance of tissue engineering, I should provide a definition of tissue engineering. The tissue engineering is the study of biological and engineering methods to improve or substitute certain tissues. While tissue engineering is a relatively nascent field, it has been rapidly expanding due to the immense need of this field. Tissue engineering can greatly improve current medical practices by replacing diseased tissue. These medical practices tend to require organ donations, which are scarce. According to the United Network for Organ Sharing, one person is added to the transplant waiting list every 10 minutes, and approximately 20 patients die each day due to the lack of transplants. Other treatments such as autografts, allografts, xenografts, and prosthetics have side effects such as immune rejection, multiple surgeries, lack of proper function, and shortage, which makes developing new tissue from tissue engineering a valuable resource. Another application is the development of tissue models for research. Once again, due to the shortage of proper tissue, it becomes difficult to test new products such as drugs or biomaterials. Tissue engineering provides an avenue for new tissue models for research. When planning a tissue engineering study, there are three major components to consider. These include the cells, scaffolds, and the signals. This diagram indicates that successful tissue engineering construct requires an effective integration between the three components, and these three components will be discussed later. Cells are the principal constituents of the developing tissue. If the tissue engineering construct is successfully integrated, then the cells will comprise most of the replacing tissue. The major cell source for tissue engineering are stem cells, due to their potential to differentiate into several types of cells. Mesenchymal stem cells are the most abundant stem cells, and these stem cells can be found in adults, mostly in the bone marrow. However, these cells can only differentiate into blood vessel, bone cartilage, muscle, and fat tissue, limiting their potential in tissue engineering applications. The figure shown features the differentiation of mesenchymal stem cells into the different types of tissue. This differentiation is mainly controlled by signals, which will be explained later. Another type of cell is embryonic stem cell. These cells can differentiate into many more cell types. However, the embryonic stem cells are scarce, and obtaining these cells from embryos creates ethical controversy. A third cell source is induced pluripotent stem cells. These cells are originally somatic, but is reverted to a pluripotent state by genetic manipulation such as through transcription factors. These iPSCs have the pluripotency seen in embryonic stem cells, but has a more abundant source seen in mesenchymal stem cells. However, because this technology was only developed about a decade ago, not much is understood on producing iPSCs, and a conversion from somatic to iPSCs are currently not effective enough for clinical practice. The second component of a tissue engineering construct is a scaffold. It is the structure used to guide the development of the tissue. The scaffold should provide a surface for cells to adhere and migrate for the development of the tissue. After development, the scaffold should dissolve to allow the newly formed tissue to fully integrate into its surrounding. The scaffold can be formed from several types of biomaterials such as ceramics, natural polymers, and synthetic polymers. Synthetic ceramics such as calcium and hydroxyapatite are hard and stiff, making them applicable to bone formation. Natural polymers such as collagen and silk provides easier cell adhesion and migration, but might be hard to produce for a scaffold. Synthetic polymers such as polycaprolactone and PLGA are abundant, but might not provide the cell integration fully. The final components are signals, which are molecules that instruct cells to differentiate and develop into a certain type of tissue. These molecules trigger certain signaling pathways of the cells, which eventually progresses into differentiation and cell division. They prevent the formation of a different type of tissue or overgrowth, such as the case of teratomas, a tumor consisting of cells of different types. For example, in the figure seen in an earlier slide of mesenchymal stem cells, These certain signals can lead the MSC to differentiate into a certain cell type.